You got a screensaver?
Yeah. All right, guys, girls, and guys. So, let's get ready to science a little bit, the science review. So, we're going to cover units 2, 3, 4, and 5 really quick like, okay? So, if I need to stop me. So, if you have a question. So, let me get your attention up here. Let's pull the mask up off my eyes, Lord. Let me get your attention up here so we can make sure we're straight. All right. So unit 2 covers the characteristics of life, okay? And so we stated there were 8 things that you needed to know about in order to classify something as living. What's the first one, the most important one?
You need to have a cell.
Made of one or more cells. What else?
Yeah.
Maintain homeostasis. What else? Genetic material. What else?
Grow as a group.
Grow and reproduce. Grow and develop and reproduce. So that's respond to changes in the environment.
Take energy.
Yes, use and obtain energy, and they are able to evolve. Those are the 8 things.
Good job.
That you guys need to make sure you understand. Now, the cell theory has 3 tenets or 3 parts. Okay, the cell theory states 1, that the cell is the basic unit of structure and function for all living things. 2,
they're made of one.
All living things are made of 1 or more cells. And 3, all cells come from pre-existing cells. We have the cell theory because of the work done by three different scientists in particular. 4 different scientists in particular. Which scientist is credited with giving us the information that led to the discovery that the cell is the basic unit of structure and function for living things? Hooke. That was Hooke. Which scientist or scientists, with the plural, is credited for the part of the theory that states that all living things are made of 1 or more cells? Schleiden and Schwann. Schwann said the animals, Schleiden said the plants. Together, that's all living things. All right, and which scientist is credited for giving us the information that cells come from pre-existing cells?
I don't remember his name.
Virchow. All right, so viruses. Are viruses classified as living or non-living?
Non-living.
Why?
Because they don't have cells.
They don't do all 8 of those things, right? They don't have cells. They don't reproduce on their own. They reproduce, but how?
In
a host cell. They do have genetic information. They do evolve, but they don't respond to changes in the environment. They don't have the organelles to obtain and use energy. So viruses do not do all 8 of the characteristics of life. So viruses are considered non-living. Even though they have some of the characteristics, they don't have all of the characteristics. Now that was unit 2. Any questions about unit 2?
What were the 3 cell theory you said? They need to come from pre-existent cells. All living things are made of cells.
And the cell is the basic unit of life. All right, so that was the tenets of the cell theory. All right, unit 2 talked about the major organic compounds. I don't know how to split this thing.
You want to help?
Can you pull that side up a little?
I can help you.
Oh,
Break
great.
it.
It's upside down when I flip it.
Didn't you need to
Yeah, I need to
that?
turn it around. When I flip it, it's upside down. Woo! Ow. Help a sister out.
Can you?
Parker, somebody. Help a sister out. Help. Help me. All right.
These things are the same no more.
All right. In your note packet, don't forget, at the end of that unit, there is a table. This is unit 3. You need to make sure you can fill out that table. This deals with major organic compounds or carbon-based compounds that are essential for life. We talked about the fact that something that is organic contains carbon. Carbon makes 4 covalent bonds, so it is the center of this. It likes to bond to other carbons, hydrogen, oxygen, and nitrogen. We talked about how monomers join together. When they join together, water comes out. So that's what type of synthesis?
Huh?
When they join together, when monomers bond, water comes out. So that's what? Dehydration. To break them apart, water goes in, and that is hydrolysis, okay? So when the parts of a compound join, that's a dehydration synthesis for these macromolecules. And when the monomers break apart, that's a hydrolysis reaction. Now, the 4 major organic compounds are carbs, lipids, proteins, and nucleic acids, okay? Carbohydrates are made up of three elements: carbon, hydrogen, and oxygen. What is the monomer for a carbohydrate?
Monosaccharide.
Monosaccharide. What does monosaccharide mean?
Glucose.
One sugar. Glucose is an example. Sucrose is an example. Okay? Most sugars, especially if they are monosaccharides, end with ose. All right? Now, the polymers, we talked about specific polymers. The specific polymers we talked about were found in the plants or animals. What were the two found in plants?
Starch and
Cellulose.
Cellulose.
Starch and cellulose in plants. What about animals?
kitchen and glycol.
Chitin and glycogen. Chitin and glycogen, okay? Starch and glycogen are used for storage. Chitin and cellulose are used for structure. But what is the main function of carbohydrates?
Short-term energy.
Short-term energy storage. Here's a little tidbit for you. Your brain can only use those monosaccharides to actually function. So it needs the energy from monosaccharides.
Wow.
So keep that in mind.
So I need
All
to-
right. Now, the next one was lipids. Is a lipid a true polymer, though?
No.
No, because it's made up of different parts, okay? The parts that make up a lipid are going to be
Carbon.
either if it's a triglyceride, it's a glycerol and 3 fatty acids. If it's a phospholipid, it's a glycerol, 2 fatty acids, and a phosphate. If it's a steroid, there's 4 fused hydrocarbon rings, okay? So the parts that make up a lipid are different. What lipids have in common is that they are water insoluble, which means they are hydrophobic. They do not react with water. They're non-polar, okay?
I have.
Yes.
What if phosphate half of it is hydrolipids and
Let me finish. Let me finish. All right. Now, the elements that make up triglycerides are carbon, hydrogen, and oxygen. The exception is the phospholipid, which also has phosphorus in it, and that phosphorus is found in a phosphate group. A phosphate is a polyatomic ion, which means it carries a charge. And remember, like dissolves like. Water has a partial charge. Phosphate group has a charge, so they interact with each other. The phospholipid is special because half of it is hydrophilic, the part with the phosphate head, but the part with the fatty acid chains is hydrophobic.
which you like.
There's 4 major types of lipids: triglycerides, phospholipids, steroids, and wax. The triglyceride is the main type. That's what we call fats. Triglycerides can be saturated or unsaturated, and their function is long-term energy storage. Your phospholipids are amphipathic, where half of it, the head, is hydrophilic, the tails are hydrophobic. Their function is to make up the plasma membrane, the cell membrane, okay? And steroids, we can identify because they're made up of 4 fused hydrocarbon rings. An example of a steroid would be cholesterol, and they can be used for chemical signaling. To build those steroid hormones. Cholesterol can also be a part of the cell membrane to help the membrane maintain its fluidity or its ability to move properly. Wax is an honorable mention because I like to bring up wax. It's also one, and it's used as a protective covering to prevent water loss, okay? The next one was proteins. Proteins are made up of carbon, hydrogen, oxygen, and nitrogen. Proteins have a monomer called what?
Amino acid.
Amino acids. An amino acid has an amino group at one end and a carboxyl group at the other end. That carboxyl is called a carboxylic acid. That's why it's called an amino acid. The backbone is the same for all amino acids. They only differ at the R group. That's what they vary. There's 20 different amino acids. The polymer is called a polypeptide. There's several different examples of proteins. You can think about muscles. You can think about transport proteins in the membrane. You can think about the proteins that function in your immune system. There's several different functions. One of the important functions is that some proteins function as a catalyst. We call those enzymes. Enzymes are a special type of catalytic protein. Their job is to decrease the activation energy of a reaction so the reaction can get going faster, okay? So that's what an enzyme does. And most enzymes end with ACE. Enzymes are also specific for what their substrate or the thing that they react on, they bond to for the reaction. For instance, sucrose will be used with an enzyme called sucrase. But lactose has a different enzyme. It can't use sucrase, it would have to use lactase, okay? As I said, there's several different functions and several different examples for proteins. Nucleic acids are our last major organic compound made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So those 5 key elements make up a nucleic acid. What's the monomer for a nucleic acid? Nucleotide. A nucleotide has 3 parts. The sugar, which is a 5-carbon sugar, either ribose or deoxyribose, a phosphate group, and a nitrogen base. That nitrogen base could be adenine, thymine, guanine, cytosine, or uracil. 1 of 5 nitrogen bases. The polymer is called a polynucleotide. The 2 examples that we talked about are DNA and RNA, but don't forget ATP is also a nucleic acid because it has the same 3 parts. It's an RNA nucleotide with 2 extra phosphate groups. We'll talk about that in the next unit. Functions: store and transmit genetic information and make proteins. That's what will direct the synthesis of proteins. They don't actually, the ribosome itself makes the proteins, but it directs the synthesis of proteins. That's unit 3. Anybody have a question about unit 3?
No, man.
Make sure you know all you need to know about those major organic compounds, carbs, lipids, proteins, nucleic acids, and also about enzymes and their specificity. Enzymes are specific for their shape of the substrate, the pH, the temperature, and the salinity that they work in, and also the concentration, okay? If you change the pH, temperature, and salinity, the enzyme will begin to denature or unfold, and we know that structure dictates function. So if you change the structure, you change the function. All right, that was unit 3. Unit 4, cell organelles, okay? Make sure you know your cell organelles. What's the function of a mitochondria?
Aesthetics.
Makes ATP. What about chloroplast? It does what? It does photosynthesis, which means it takes light energy and stores it in chemical energy in sugar. Mitochondria and chloroplasts are what we call energy transformers. Chloroplast transforms light energy to chemical energy in food. Mitochondria transfers chemical energy from food to ATP. They're energy transformers. All right, what about the nucleus?
Nucleus, it gives control, commands.
Controls most of the cell's activity. We say most, not all, because DNA is found in mitochondria and chloroplasts as well. They have their own DNA, okay? Most of the cell's activities are controlled by the DNA in the nucleus. What's the job of the ribosome?
Makes protein.
Makes proteins. What about rough ER?
Rough ER, it's rough.
Transport the proteins that are going where?
Outside the cell.
Outside the cell. What about smooth ER? Make and transport lipids, okay? Don't forget that. And it also detoxifies chemicals.
Oh yeah, I remember that.
Golgi. Packages, sorts, modifies proteins and lipids that are leaving the cell. Cell membrane. Controls what goes in and out of the cell. That's how it helps to maintain homeostasis. Lysosome.
Cleans the trash.
What kind of cells? Animal or plant?
Plant.
Animal. Vacuoles.
Takes the thing.
Storage of water and waste. What about cell wall? Structure and support. All right, remember the 3 C's. Things that are found in plant cells but not animal cells. Chloroplast, cell wall, central vacuole. That's a very large vacuole. You'll find those in plant cells but not animal cells. Things found in animal cells but not plant, lysosomes, centrioles. All right.
I thought there were 3.
Is it 3?
Yeah.
Yeah, what's the third one?
I don't remember.
Me either. You better cover your mouth and your nose up.
You know what nemesis.
Huh? Chloroplast is found in plant, not animals. Yeah, he was talking about animals, not plant. All right. Dang it.
Look it up.
No. All right. That's unit 4. Any questions about unit 4? You need to go over your cell organelles again. Unit 5. Let me turn this back around. Move your feet. Move your feet. Hey. Move your feet. Hey. Move your feet. Move your feet. All right. Unit 5. All right. Cell transport. Is this talking about transport across the plasma membrane? Push this back some so you can see. Transport across the cell membrane. The cell membrane is mainly phospholipids. There's proteins embedded in or attached to it. There's also cholesterol, and on some of those proteins, you have carbohydrates attached to those for cell communication and cell recognition. Now, when something passes through the cell membrane, we call that cellular transport. There's two ways that cellular transport can happen: passive transport or active transport. Passive transport goes from the area of high concentration to an area of low, so we say it goes with the gradient. That's the natural flow of things. Active transport is the opposite. It goes from low concentration to high, so it goes against the gradient, okay? And whenever you go against something, you have to put in extra effort. So to go from low to high requires ATP, but high to low is going with the flow, so there's no energy that's required. There's 3 different types of passive transport: diffusion, facilitated diffusion, and osmosis. Diffusion is simply when small and nonpolar things can go straight through that lipid bilayer. Things that are large or charged have to go through a protein. They need help to cross the bilayer. They can't go straight through it. They have to go through a protein, the transport protein. This is called a facilitator because it's helping them. So this is called facilitated diffusion. Okay, they're going from high to low, but they're going through a protein. And that happens if you're large or charged. That means polar or ionic. Osmosis is the movement of water. Water goes through these things called aquaporins, special pores, from an area of high concentration to low concentration. So a cell in its environment, water can move one of 3 ways. It can go from outside of the cell into the cell, in the cell to out of the cell, or in and out at the same rate, okay? Now, if the cell has less water than the environment, because water goes from high to low, water will move into the cell. That's called hypotonic. Cell swells, get big like a hippo, hypotonic. If the cell is sitting in, has more water on the inside than the environment does, then the cell loses water because water goes from high to low. So that's called hypertonic, and the cell will begin to shrink. If the concentration of water is the same inside and outside of the cell, the water goes in and out at the same rate. Now let me clarify something. In hypo and hyper, water's going in and out. It's going one way faster than the other way. Okay? All right. Now, when it comes to plant and animal cells, because plants have a cell wall that help resist that osmotic pressure, plants prefer to be in a hypotonic environment. Plants like it when more water is coming in than going out. The cell wall prevents them from bursting. Animal cells would burst in a hypotonic environment. No cells like hypertonic because no cell wants to lose water faster than it can take it in. That's death. Animal cells prefer an isotonic environment because animal cells like to resist a change in shape and size. They don't want to change their shape or size. Active transport is going to involve the cell membrane. If it's a bulk transport going from high to low, the cell membrane is either going to wrap around something and pull it in, or a transport vesicle is going to fuse the cell membrane so it can push something out. When stuff comes in, it's called endocytosis. If it brings in a solid particle, it's called phagocytosis because phago means to eat. If it brings in a dissolved particles, it's called pinocytosis because pino means to drink. So the cell has 2 ways of getting things inside, just like we do, eating or drinking. If it eats, it's phagocytosis. If it drinks, it's pinocytosis. Whenever something is pushed out of the cell, a bulk piece is pushed out of the cell from an area of low concentration to an area of high, we call that exocytosis, all right? And that is it on Unit 5. Any questions about that?
Okay.
You said I ain't talking to you till you cover your nose and your mouth.
Okay.
What's up?
You said phago is for eat it, right?
Mm-hmm.
And pano is for
Drink.
Drink. But phago is a drink.
Pino.
Phago is a drink.
Yeah, but I said phago, not phago.
But phago. Oh.
not Faye, go.
Oh, what about peanuts?
That's P-I, not P-E.
They
Why you
should
trying to be
switch the
funny?
names. They
Uh-huh.
should switch the names, right?
No, they should not.
Yeah.
All right, anyway, I ain't thinking about you right now, my hum. Any other legitimate questions? All right, guys, I recorded this. Hopefully my phone didn't die because it did say low battery before. It didn't die? Cool
Nope.
beans. All right, I recorded it. I'm getting ready to upload it into Ed Puzzle. And your homework is going to be to rewatch the review. All right, bye.
I have a question.
What's up?